Good afternoon, everyone, and welcome to the College of Engineering Virtual Brown Bag Series. I'm Gautam Das. I'm the Associate Dean for Research in the College of Engineering here at UTA, and I'll be the host for today's presentation. Today's presentation is going to be titled Self-disinfecting Nanocomposites to Prevent Infections, and the presenters are from our very own college. Luis Soto-Garcia, who is a doctoral candidate in bioengineering department, and this is his main research topic. And his advisor, Kytai Nguyen, is also going to be there. She is a professor in the bioengineering department, and she'll be also there to assist in the presentation. So a brief abstract about what's going to happen. This pathogens of bacterial and viral origins cause significant infections in humans and have a large impact on public health. And it's very important for preventing contagion, especially in this day and age when there's a lot of resistance to antibiotic resistance and the lack of treatment, lack of vaccines, and so on. So this research is going to be focusing on how we can create self-disinfecting nanocomposites. So I will not go through the rest of the abstract. I will rather leave it to the presenters to talk more about it during the talk. Very, very exciting work. A little bit of bio about these presenters. The professor, who is Dr. Nguyen, she received a bachelor's in chemical engineering from the University of Minnesota in 1995. And a PhD in chemical engineering with an emphasis on bioengineering from Rice University in 2000. And she's currently a professor in the bioengineering and nanotechnology track, advisor at the University of Texas at Arlington. Her research interests include vascular tissue engineering, nanotherapeutic strategies for vascular and lung regeneration, and nanoparticle, microparticle, and injectable hydrogels for controlled drug delivery and tissue engineering applications. She also works on nanoparticles for detection and treatment of various diseases, including cancer, cardiovascular diseases, and lung disorders. Her PhD student, Luis, who is going to be the main presenter today, he obtained his BS in chemical engineering from Instituto Tecnológico de Durango in 1984, and MS in chemical engineering from the Universidad Autónoma de Nuevo León in 1986. After 15 plus years of working in the chemical process industry and 15 years as a bilingual teacher, so a very experienced person in lots of different disciplines, he's currently pursuing a PhD in biomedical engineering with Dr. Kytai Nguyen. He is also interested in the unique abilities of blue mussels to adhere to different surfaces under wet conditions, and as a result, he's developing biomimetic adhesive nanoparticles for different bioengineering applications such as drug delivery, tissue engineering, or antimicrobial materials. His future work includes the use of nanocomposites to create self-disinfecting materials to prevent pathogen infection and develop an angioplasty balloon coating for drug delivery and treatment of peripheral artery disease. So with this, I would like to welcome the speakers to start giving the presentation. A couple of suggestions for the audience. So the talk is going to be going on for about 40-45 minutes, maybe a little bit more. Please, if you have any questions, please type them in the chat windows that is available, the Q&A windows. And once the presentation is over, we will use the remaining time where I will read out these questions to the presenters and we will get their answers. So with this, let me welcome Dr. Soto-Garcia. The PhD student, Dr. Soto-Garcia, to give the presentation. Not yet doctor, but hopefully will be soon.
Thank you, Dr. Das. Thanks everyone for being here for this presentation. So our topic is regarding infections in this era of health problems. What we're gonna learn today is a little bit of background on infections. We will discuss a little bit about the ways of disease transmission, current approaches to prevent infection, specifically passive infection, Then the approaches that are based on nanotechnology to fight these problems. Some antimicrobial strategies, then we will review very quickly the theoretical base for my research, and then I will go over what are we currently doing in the lab. So let's begin with a little bit of background. We know that microbes are all around us. Basically, virus and bacteria, we can find them on us, within us, and around us. And the good thing is that the vast majority of these microorganisms are harmless. They just are there. They don't do any harm to us. However, it is important to notice that for instance, in our microbiota, we have to have microbes in order to process food, etc. But there are some of them that are called pathogens. There are the list of the microbes that they can cause diseases. Scientists have identified about 1,400 human pathogens. And these pathogens are related to most common causes of mortality. And actually, the human history, our own history, has been shaped by pathogens, by our evolution, our demography, migrations, our history is closely related to these human pathogens. Now let's talk a little bit about infections and disease transmission. There are two main routes of infection. One is the direct route, the other is the indirect route. For instance, in the case of direct route, one example would be bites. If you get bitten by a dog, for instance, you can get some kind of disease. That would be an example of direct contact transmission. Also, we can have another example of direct transmission when someone sneezes close to you or over you, and then you get infected as a result because of the droplet spread. So those are classical examples of direct route of infection. We also can get infected via indirect routes. For instance, aerosols. Now that we have in the pandemic with COVID-19, scientists have discovered that aerosol is the main route of infection. That would be an airborne route of infection. But also we have another indirect routes like vehicle-borne routes. We can get infected through food, through water, or through fomites. We will discuss a little bit more about fomites in future slides. So just keep this word in mind, fomites. And another way to get indirect transmission is through vector-borne. In this case, for instance, mosquitoes, West Nile virus here in Texas. So these are basically the routes of infection. And I would like to focus on this word fomites because that is directly related to my research. And the question here would be, what are fomites? Fomites, think about inanimate objects. They can be easily contaminated by an infected individual. For instance, the table where I have my laptop is a fomite. It's inanimated, but if I were sick, I could easily deposit on the surface of this table the droplets or any kind of germs from my hands. So any inanimate object that can be contaminated by an infected individual is a fomite. Clothes, personal protection equipment like the coat that I'm wearing, frequently touched surfaces like doorknobs or the touch screen of your cell phone. Those are classical examples of fomites. And fomites can become a passive route of infection. We say passive because they are not actively infecting us. But in order to get infected, we have to touch them and then touch our heads, our faces, and transmit the pathogens through that secondary route to ourselves. And it's important to notice that fomites are an interesting subject because pathogens can survive on them for extended periods of time, especially if fomites are non-porous. For instance, think about stainless steel or aluminum or glass. In those cases that are not porous, the pathogens can live between few days to as long as some months. So they can become, the fomites can become a very important way of transmission. Now the natural question could be, how frequent is the infection via fomites? In the cases of influenza, rhinovirus, or norovirus, it has been identified as a really important route of infection. But also scientists have discovered that entire microbial communities and antibiotic-resistant bacteria can be transmitted indirectly via fomites. Think about a room in a clinical setting. There is a lot of surfaces that people touch very frequently. Those surfaces can become fomites that are sources of these resistant bacteria, like MRSA, for instance. So in this diagram, we can see the different sources of infection for fomites, but basically they come from humans. The pathogens come from humans, are deposited on surfaces. Then by touch, we can touch the surface and then touch our faces and get infected by ourselves. So that's the mechanism of infection via fomites. And as I previously mentioned, it's a secondary way of infection. It's passive because the pathogens are there, but we have to touch the surface in order to get ourselves infected. So let's discuss now about the current approaches to prevent this kind of passive infection. There are some recommendations to prevent the passive infections. Frequent cleaning and disinfection of surfaces is the most used. We can see that there are many EPA-approved products like quaternium ammonium salts, but also in absence of EPA-approved products, we can disinfect the surfaces using 1% bleach or 70% ethanol. For instance, if we talk about our clothes, we can wash them in heated cycles. And it is also recommended that we disinfect laundry baskets or any kind of surface that can get contaminated with pathogens. There are a little bit more sophisticated methods of controlling the passive infection. For instance, the use of UV light or the use of peroxide vapors, but they require more equipment than just the cleaning of the surface. And besides these methods have an issue, have some limitations. In this case, FTS stands for frequently touched surfaces. Frequently touched surfaces can be recontaminated any moment after we apply the disinfectants. If we clean up a table and a few minutes later a person sneezes around the table, then the table is going to get contaminated again. So in the case of frequently touched surfaces, they need the constant application of disinfectant, the repeated application. In the case of personal protective equipment, PPE, the equipment doesn't have a system to inactivate the collected pathogens. Think of a face mask. If you have your face mask on, it's going to protect you by collecting those pathogens on the outer surface. But there is no system to kill or to inactivate those pathogens. So if you later touch the surface of the mask and then you touch your face, you're going to get a secondary infection as a result of the inadequate use or the disposal of the mask. Besides washing personal protective equipment, especially respirators like N95 or face masks, other than cloth masks, sometimes is not possible without damaging the respirator. So those can be the main limitations of the current technologies. So we can say that there is a gap. There is an opportunity here to improve. Sportelli et al., they prepared a letter at the beginning of the pandemic and that they submitted to some publishers. And they basically were questioning and identifying this gap. And basically, they mentioned that we need to do more effort to increase the efficacy of personal protection equipment and to develop more efficient and synergistic antiviral coatings. So based on this paper that was published at the early days of the pandemic, we in our lab, we began to try to figure out opportunities to help close this gap in infection prevention. As you can see in this diagram that is included in that publication from Sportelli, besides the focus on finding cures for the problem, specifically for the COVID-19 problem, besides working on antivirals, etc., they also propose to work on improving the personal protection equipment, respirators, and also developing antiviral cleaning surfaces. So based on this information, we have been working, and there are some approaches based on nanotechnology to address this issue, this problem. But before I continue to discuss the specific approaches of nanotechnology, I would like to make a pause to talk a little bit about what is nanotechnology. Basically, nanotechnology is science, engineering, and technology conducted at the nanoscale. But here you could say, okay, what is the nanoscale? And that's a really interesting question because we are talking about really, really small things. So nanotechnology is the study and application of extremely small things. And it can be used across all science fields. I will give you an example of nanotechnology in the next slide, but first I would like to recognize Richard Feynman. He's known as the father of nanotechnology because he was the first one that began talking about the nanoscale and the importance of this scale in the whole context of science. So the question would be, how small is nano? What is nano? So nano is the scale at which much of the living biology occurs. Think about DNA, for instance. DNA is very small, it's around 3 nanometers in diameter. The SARS-CoV-2 virus is between 60 and 140 nanometers in size, in diameter. And if you think of a piece of paper is about 100,000 nanometers thick. So nano is something really, 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 really small. When I was looking for materials, trying to explain how small is nano, I found some really interesting videos, and I'm gonna reproduce them for you, like this one, Understanding Nanoscale.
Luis, I think your video is frozen.
I think we may have lost Luis himself.
Luis, I think you may skip the video.
Dr. Doss, can you talk for a minute, or have Dr. Nguyen talk for a minute? Well, he had to drop out, I guess, and he had bad connection. He'll be back in.
Yes, I think at this point, maybe we can ask Dr. Nguyen to maybe try to fill up a little bit of what, the interesting video that Luis was talking about.
Yeah.
We'll hope we can get him back soon.
Mm-hmm. I think nanoscale, what he want to show you is very much in the scale, very small site. In the thing we compare of the marble with the earth, and then you can see that the red blood cell is about 7 micrometer, and then later on we have several DNA, RNA, though a very small scale. So nanotechnology is very small scale that we develop the technology in a very small scale that will be adapt with the in our body, in our cell. It will why we have a technology is more effective, because it will interact with the cell, easily uptake by the cell. So that will be another thing. Let I see if I can share the screen and continue the presentation. I had to open the I had to open my file. And share. Sorry for the technology. Technical issue.
No problem. It happens once in a while.
Okay, let's see. Okay. If Luis cannot finish, then I will try to share my sprint. Okay. Okay. Can you see my screen right now?
We can see it, but can you put it in slideshow? There you go. Perfect.
Okay, good. So you can see my slideshow, right? Let I go back to the essential question. Can nanotechnology help the fight against pathogen, and how? So we have nanotechnology have been used for against the infection. However, the topic is not explored enough until recently. The nanotechnology have been do prevention, diagnosis, and treatment. However, for the infection, we have been developed some technology, but not explored enough. So based on the current technology, we think that the FTS and PPE effectiveness could be enhanced by including nanotechnology-based antimicrobial. Because in our research, we have developed many nanomaterial that consist the antimicrobial property. So the nanotechnology-based proactive antimicrobial system can be important to prevent the infection, especially on the formite, on the surface touched by the infected patient. So we have several antimicrobial strategies. For example, we can modify the surface with antimicrobial coating. For example, we can use different type of agent, active eluting agent, immobilized molecules, like active molecules. And for the, or we can use the super hydrophobic nanostructural surface. We can also do the contact active surface, biocide releasing surface. So those strategies have been used in order to revive antimicrobial property to prevent the pathogen, the infection. So here is our strategy. So our strategy shown this image, you can see that we utilize activate biorelation surface. How we do that? We develop the dopamine-based adhesive hydrogel. And this one, dopamine, is mimicking muscle adhesive property. So this hydrogel will adhere to the surface very tightly because it mimics the muscle adhesive property. You know, the muscle is adhere on the wet surface very strongly. So this material is dopamine-based adhesive hydrogel, so it will stuck on the surface very strongly. And include with this material, we develop a photocatalysis metal oxide or PMO. So what does mean with this nanoparticle? You can use light to activate, and then it will release the reactive oxygen species. It will release something that can inhibit the growth of the pathogen. So it can kill the virus, it can kill the bacteria when you shine the light. So by that, when you will mix this with the hydrogel, we will reduce the biocidal nanocomposite. And this nanocomposite will adhere on the surface, and when you shine the light, it will inactivate all of the pathogen on the surface for a long time. So we can use this as a surface coating or as the PPA modified. So by this, we can develop of our strategy. We can show that, for example, the patient will be sneezing and then develop the pathogen on the surface. So on this surface, we will shine the light and it will reduce the reactive oxygen species. And this reactive oxygen speed will be generated from the nanoparticle after you shine the light, and this speed will kill all of the pathogen. So it will make it inactive all the time when you hand the light shine on the surface. So this will, we call this material is self-disinfecting nanocomposite, because when you just shine the light, it will self-disinfecting. It will kill the pathogen, virus, bacteria continuously when you shine the light on the surface and coating with this material over the time. So you don't have to apply material like bleach. You have to apply several times, or you use solution, disinfectant solution, use all the time on the surface. You just coat it one time and shine the light, and it will be stay there for over the time. So our material based on both photocatalytic effect and reactive oxygen species. So what does it mean with photocatalysis? When you shine the light, it will activate, it will disinfect the pathogen. It will kill the pathogen over several mechanisms. For example, titanium oxide. Titanium oxide, when you shine the light, the photocatalyst will be on the photo absorption from the UV light, and then it will move the electrons, and then it will dissipate the energy at the heat, and then it will convert it to the oxygen radical form or reactive oxygen species. And this reactive oxygen species will kill the bacteria or the virus. So this is the principle of photocatalysis. Let's see, I really want to see the video, but I don't want to frozen this and get kicked out of the system. Let's see if I can do it. I want you to show the principle of photo. Can you hear? Okay. So let I continue with our strategy. So that titanium oxide can be sulfate modified with the dopamine. And then when we sulfate modify with the dopamine using the in situ polymerization. So for this modify, then it can be when the light, visible light, stimulate on this coating, it will reduce the reactive oxygen species. And then from this reactive oxygen species, it can kill the bacteria, the virus, and it will facilitate the titanium oxide photocatalysis under the visible light. So that is one of the strategy of our current research. So the goal of our current research is to use the nanotechnology to develop a visible light activate self-disinfecting nanoparticle or nanocomposite. We will use this material to coat on the surface, frequent touch surface, or to integrate in our protective equipment, or provide the proactive antimicrobial capability. So for that, we develop our hypothesis Thus, we had previously developed bioadhesive material, bioadhesive nanoparticle adhesive hydrogel, like responsive nanoparticle made of photocatalytic metal oxide. So our hypothesis that our nanoparticle PMO nanoparticle can reduce the oxygen radical, and this reduced oxygen radical can kill the bacteria and virus, and it will have a very high antimicrobial property. So our long-term hypothesis that our cell discipline nanocomposites formulate with the hydrogel and PMO nanoparticle can form cell disinfectant coating with effective and long-lasting biocidal property triggered by visible light irradiation. So for this, we already show you how the material coats on the surface can release the reactive oxygen species after you shine the visible light, and it will keep the pathogen inactive when it touches the surface. So it will kill all of the pathogen for our material. So in order to do that, we have three specific aims. We have formulation and characterization and optimization of DMs. We also have in vitro study to evaluate of the antibacterial effect of this formulation. And then we have in vitro study to see if this material is safe to use for human use. So the innovative aspect, we had the material will use visible light to kill the pathogen. It will help to disrupt the formite disease transmission pathway. It easily to formulate, and also it environment friendly. It maintains the property, the biocidal property for about 60 days. So we had this material will have significant and impact to treat many diseases, for example, including the COVID-19 recently. For our preliminary study, we had developed the adhesive dopamine arginate dopamine polymer, and you can see this from this one when you had dopamine and incorporate with the hydrogel called arginate. We can see that it involved with the CC aromatic stress and also on the peak of the dopamine. And we also find out that the arginate dopamine and also our nanocomposite material is more stronger compared to the arginate, the hydrogel alone.
Dr. Nguyen,
Yes.
I'm back. Fortunately, can I keep going? If you you can keep moving the slides. We also have been working with hyaluronic acid hydrogels, and in previous research we demonstrated that these hyaluronic acid hydrogels are really efficient in addition. Here we have the FDR spectra and the different studies on adhesive strength. Can you please go to the next slide?
Good. Let I go.
Thank you. Also, we have been working with the development of nanoparticles. In this particular case, we prepared nanoparticles of polydopamine. Just remember that in our design, we're going to be using polydopamine. And these polydopamine nanoparticles, we confirmed that they are attached to, they are polymerized according to this spectra. And in the image to your right, you can see a transmission electron microscopy image showing these nanoparticles, polydopamine nanoparticles. Can you please go ahead, Dr. Nguyen? We also worked on understanding the antimicrobial properties of these hyaluronic acid hydrogels, and we found that they were able to kill Escherichia coli and S. aureus, which is a base for our next studies. Can you please go to the next one? So just remember that in our hypothesis, we are going to combine a hydrogel based on dopamine with titanium oxide coated with polydopamine to prepare the self-disinfecting nanocomposites. Please go ahead, Dr. Nguyen. So our nanoparticle design includes a core of a photocatalytic metal oxide, which is the titanium oxide, and the coating is going to be the polydopamine. And the purpose of the coating is to make the titanium oxide able to form reactive oxygen species under visible light irradiation, which is the more innovative part of our design. Can you go ahead, please? These are our current studies. We have been working on developing a hydrogel that is adhesive, and it is based on fucoidan. Fucoidan is a material that is extracted from brown algae, and by itself, fucoidan is known as a material that impedes the development of microbial coatings. So it's an antifouling material besides other properties. So we formulated a hydrogel with fucoidan dopamine. We confirmed the conjugation, as you can see in the image with under the letter A. And we have been preparing these materials with concentrations about 35% of dopamine. And on the pictures on C, we deposited a droplet, a sample of the material onto an aluminum surface to observe the way it adheres. And we first observe a very low contact angle, which is an evidence that the material is spreadable and can easily get in contact with the surface. And later, after 48 hours, we observe the formation of a film. And in this case, the brownish coloration of the film is an evidence that the dopamine that is content in the hydrogel is cross-linking. In other words, is reacting and forming links to adhere between the dopamine molecules and also to the aluminum molecules on the surface. Can you please go to the next one? This is a short video, but can we skip it just to avoid any issue?
Okay.
Just in case.
Okay.
It's just a video where it shows what is called the tap and peel test. Well, basically, we just put a piece of tape on top of the hydrogel, and then we quickly remove the the tape, and we observe if is there any kind of dislodging of the material. And this is a quick qualitative test. Of course, later on we will perform more studies, more on the quantitative aspect. But in this case, the material was able to pass the test. We also tested our material on antimicrobial properties, and we found that our hydrogel made of fucoidan and dopamine can kill S. aureus and E. coli. We determined the minimum inhibitory concentration of these materials. And in the next slide, we're going to see, can you please go to the next slide, Dr. Nguyen? Thank you. Here we did some studies on the time required to kill the bacteria, and we found that this hydrogel by itself can kill bacteria in a period of between 8 and 12 hours of exposure. Can you please go ahead to the next one, please? Then we began preparing our photocatalytic nanoparticles, and we determined to your left, you see image A. That's the sizing of this nanoparticle. The size is around 200 nanometers. And to the right, you see the spectral image that confirms the formation of the coating on top of the titanium oxide core. Can you please move to the next one? And then we use rhodamine to test the formation of free oxygen or reactive oxygen species. And in this case, we expose rhodamine, which can decompose under the action of oxygen, as a way to check if our material was generating this oxygen species. And on your left, on the graph A, you can see a peak. That peak is when rhodamine is in initial conditions. And then you can see how the peak reduces until it totally disappears. And on the picture on your right, letter B, you can see the material before the treatment in the pink color and after the treatment when it becomes totally clear. That's an evidence that the action of the nanoparticles was effective on decomposing the rhodamine. After this experiment, we still had to answer the question, okay, is this going to work on bacteria? So our next experiment, we did the same thing. We exposed E. coli to our nanoparticles in a suspension. And we found that in this preliminary antimicrobial test, that nanoparticles were able to eliminate 99.999% or a 5 log reduction of bacteria, which is really good because the results for our nanoparticles were basically the same as the positive control, which was bleach added to the bacteria. So this is the last experiment that we have been conducting. Future work, we will keep working on characterizing our nanoparticles. We still need to determine the morphology using transmission electron microscopy. We need to verify the crystallographic structure using X-ray diffraction. And most important, we need to quantify what is the amount of reactive oxygen species that our nanoparticles generate under the exposure of visible light. Also, we will need to work on the optimizing of the system. For this, we are going to use a factorial design. And also, we will characterize the nanocomposite by the chemical composition, the morphology, and last but not least, we still will measure the adhesiveness of the material. And once we conclude those studies, we will keep working on optimizing the antibacterial effects on S. aureus and E. coli. There is a lot of work to be done on this optimization of the antibacterial effects, and we will aim to conclude evaluating the safeness of our materials using hemocompatibility and cytocompatibility studies. It is interesting that there are some trends for this kind of materials, not only our research, but in general, these antimicrobial coatings. For instance, some people has proposed that these materials could be embedded in the non-woven fibers to manufacture personal protective equipment. So just imagine N95 masks able to kill the bacteria on contact. So the message here is that nanotechnology has a great and extraordinary potential to help the fight against disease. But we need to invest substantial effort in order to have materials and solutions that can be translated to the clinical practice. But the field is exciting and full of discovery, and if you like to know more, please contact us.
Thank you very much for this wonderful presentation, and very timely, I must say, given that we are all struggling to keep ourselves safe during COVID times. So I'm sure that audience would have enjoyed listening to the latest research that's going on in this field. So to the audience, if you have questions, please type them out in the question answer dialog box, and when I get a chance, I'll read them out. So while that is happening, I had a few questions of my own because I'm finding this whole thing extremely fascinating, of course. You said visible light many times throughout the talk, right? I have heard of other light sources which can, kill bacteria and viruses, such as UV light, and that's been known for decades, right?
Yes.
So what is so special about visible light that this research needs to be done? Is it that visible light by itself does not kill bacteria, and therefore you have to have these nanoparticle coatings to help visible light kill bacteria and viruses, whereas UV light doesn't need all this extra help from the surfaces?
Well, what we have in mind, what we envision is a system where we could coat surfaces, for instance, in a clinical setting inside a room where there is no such thing as the sunlight. Sunlight has different spectra, UV light, visible light, etc. But inside the room, we usually have either the light that comes from the window or the light that comes from a lamp. So in this case, when we try to make the titanium oxide reactive to through the visible light, what we aim to is to create a material that can kill bacteria inside a room where there is not such thing as UV light. Of course, there are systems that use UV light inside rooms, but in order to use them, you have to leave the room, and there is to have some specific procedure to clean. But what we want to create is something that constantly works under the excitation of the visible light commonly present in a room. That would be the innovative aspect of this research.
You did mention that UV light, people have to leave the room, I guess because it's not safe for humans, and visible light is, of course, safe for humans.
Yeah, definitely.
But what about the way you're changing the surfaces? Is that safe for humans? Does the PPE need to be evaluated as to how safe it is with your modifications?
Yes, definitely. Our hypothesis is that since we are working with materials that are very common, titanium oxide is present even in food, and fucoidan is generally recognized as safe by the FDA. Our hypothesis is that since we are working with materials that are safe in nature, the product is going to conserve those characteristics. However, in aim 3 of our research, we want to validate the safeness of the materials by testing in cells in different ways to ensure that the coating is safe, because as a coating, eventually it's going to break out from the surface, and perhaps some particles of the coating can end up on some people's skin, etc. So we need to validate that those materials are safe.
Okay. So a question from the audience, how long do you think it'll take for this work to be available for public to use, that is outside your lab?
Well, I wish it was for tomorrow, given the situation, but it takes time. It takes time. For example, one single experiment might take one full week from the beginning to the completion, plus the data analysis, etc. And on top of that, let's suppose that we already had a formulation that is working. We still would have to go over all the requirements from FDA to get the material approved, etc. So it's a long-term project, perhaps a matter of some years.
Okay, interesting. So I had a question about sort of the style of research. I mean, I'm a scientist too, but I guess the kind of research you do is a little bit different from how I work. So there seems to be a lot of material science here. I mean, I see that a lot of your work involves knowing well properties of exotic materials and how they will behave in certain circumstances, right? So what led you to using the kind of materials that you decided to use to create this this particular process? I mean, I'm just trying to understand more about sort of the research procedure that you all follow.
Well, the thing is that we were working to develop the bioadhesive biomaterials for drug delivery. One of the materials that we mentioned in the presentation, based on alginate, it was developed to work as in wound healing applications as a bioglue for surgical purposes. Based on that research, we continued improving our materials. That's where we added the fucoidan material, which is known for some anti-inflammatory properties and some antimicrobial properties. So our purpose on integrating this fucoidan material was not only to add anti-inflammatory properties, but also antithrombotic properties to our materials. Because one of the uses of this material is to coat angioplasty balloons for drug delivery inside the blood vessels. So we wanted some material that would have antithrombotic properties. But as I mentioned, it also has antifouling properties. So when the COVID-19 arise, the pandemic arise, in our lab we were discussing different alternatives when we could help to fight the pandemic. And in these endless discussions, we said, okay, perhaps there is a chance to repurpose some of the research that we are doing, some of the materials that we are developing to fight different infections. And then that's when we found that the passive infection not only was in that particular time mentioned in COVID-19, but also it's a mode of transmission that is common for some other diseases. So then talking between our fellow researchers, we decided to continue exploring, and that's how we found that we could repurpose our material to make an antiviral coating that could keep its properties by itself, or in other words, becoming self-disinfecting. So that's more or less the evolution of the ideas.
Fantastic.
I would like to add more into this one. We didn't test on COVID-19, but we will send our material down to our collaborator because UTA don't have the biosafety level 3 that can work with virus COVID-19. So we will send our material to our collaborator who had the biosafety level 3 lab, and they will test on COVID-19, which will be, hopefully we will have some result next month.
Oh, okay. All right. So now there's another question from the audience. It's sort of related to the safety issue that we discussed earlier. The question is, can humans have any allergic reaction to the substance?
Well, we definitely cannot discard that effect. It could be possible, but right now we don't know until we get to the part of when we study the safeness of the material. But right now, I wouldn't discard that possibility. It could be possible.
Okay. Somebody says, thank you, very interesting research. Rosie Kelly from IMSC. She has to leave for class, but she enjoyed your presentation. I have a question, which is, so is this like a broad spectrum in some sense? Can it take care of almost all bacteria and viruses, or is it targeted towards a specific class of bacteria and viruses that and it won't work for others, or it is all very speculative right now?
Well, right now, in our studies at this moment, we are focused on bacteria, but as Dr. Nguyen mentioned, we also, thanks to the collaboration with some other universities, we will study the effects on viruses, but also in the long term, we would like to study how effective it is against fungi or some other pathogens, because eventually we would like to have some material that would give us the most protection possible. So yes, we would study in the future against bacteria, viruses, fungi, and any other kind of pathogen.
All right, so we are at the end of the hour, and while we could have continued on with a lot of interesting questions on this very, very topical piece of research, we have to bring a close to this session. Very exciting presentation. And let us thank the presenters again for spending an hour with us today. Thank you very much. And to the audience, I hope you enjoyed it, and look forward to hosting more of these.
Thank you.
Thank you. Bye.